This is Akshay over here and welcome to Akshay's biology classes. In this video session, we will try to understand the muscle contraction, specifically the sliding filament theory that explains the mechanism of muscle contraction. We shall first begin with the structure of sarcomere. In this illustration, the arrow mark shows the actin, which is referred as the thin filament, myosin, which is the thick filament, the Z line, next I band, which is the region that shows only the part of thin filaments, A band that shows the overlapping of thick and thin filaments. Next, we are having the H zone that shows the part of only the thick filaments. And finally, the sarcomere. A sarcomere is extending from one Z line to another Z line or next Z line. Let us look into the overall process of muscle contraction in general. The binding of myosin to the actin filaments, which is referred as cross-bridge formation, this results in sliding in, or more precisely speaking, myosin pulls the actin thin filaments into the center of sarcomere. The next step is detachment of myosin from the actin. This results in the filament moving back original position. So with this being understood, now we shall look into the process in more detail. The circular figure is mainly actin. The next figure is the myosin head, which is an ATPase, that is, it is having ability to split ATP to ADP and inorganic phosphate. The yellow region in the actin is the myosin binding site. So the first step mainly involves the ATP binding to the myosin head. Myosin head, being an ATPase, splits ATP to ADP and inorganic phosphate. This results in the cross-bridge formation, that is, attachment of myosin head to the actin, specifically myosin binding site on the actin. Now, the inorganic phosphate is released. This causes power stroke, that is, actin moves into the center of sarcomere. The release of ADP and binding of new ATP molecule to the myosin head will cause the cycle to repeat again and again. However, there is yet to know more, that is, role of calcium in the muscle contraction. The following picture shows the muscle cell. Next, the T tubules, also called as the transverse tubules, which are in close association with the sarcoplasmic reticulum. And finally, the neuromuscular junction, a point where the axon terminus is in contact with the muscle cell. As the nerve impulse arrive at the neuromuscular junction, the neurotransmitters are released from the axon terminus and bind to the ligand-gated channels present on the muscle cell. This generates an action potential and is transmitted into the interior of the muscle cells via the T tubules, which are in close contact with sarcoplasmic reticulum. This results in the release of calcium ions from the sarcoplasmic reticulum. We can now zoom into one section of the muscle cell showing the actin filament and actin's myosin binding site, tropomyosin, which blocks or masks the myosin binding site present on the actin. Next is troponin, which is in contact with the tropomyosin, and finally calcium. Calcium binds to the troponin that causes the troponin to shift and expose the myosin binding site, which results in the binding of myosin to actin, and the further process we have already discussed earlier. I hope this helps you all in visualizing and understanding the sliding filament theory that explains the mechanism of muscle contraction. I shall meet you people again in a new video with a new topic. Until then, thank you.